All right. Well, on behalf of the SSCD and Takeda, I'd like to welcome everybody to today's webinar, during which we will be talking about measuring outcomes and regulatory compliance. I'm Andrew Ford. I'm one of the webinar committee members and a gastroenterology fellow, and I'm just super excited to welcome our guests for today's webinar. Um, Today we will have two distinguished speakers. We will have Dr. Dan Leffler and Dr. Silvester. So Dr. Leffler is a gastroenterologist who is one of the founding members of the BIDMC Celiac Disease Center. More recently, he is working with Takeda Pharmaceuticals, Chugai Pharma, where he is currently the global project strategy lead and also working with the Celiac Disease DonQ52 program. And Dr. Silvester is a pediatric gastroenterologist at Boston Children's who is co-director of their Celiac Disease Center and extensively involved with research in that field. Uh, I just am very humbled to welcome you both, and we're excited to hear from you.
Thank you very much. So I will be starting, kicking this off, and Jocelyn will be doing the second half. Let me share my screen. Hopefully that's coming through okay. Look at, how about that? Good.
We got it.
All right, great. All right, well, thanks everyone for joining and listening to this. So by way of my background and disclosures, I am an employee of Chugai Pharma and I'm working on a celiac disease program, the DOC52 program. We won't be talking about that at all today, but goes without saying, the views uh, and concepts are mine and not should not be taken to be part of any official positions of any groups. So, all right. So, kind of a wordy title, see like these clinical trials, outcomes, and regulatory expectations. But hopefully, this will be a little bit. We'll try to make this pragmatic for whatever you do. So we split this into two sections. So I'll be taking the first sections on patient-reported endpoints and clinical trial patient selection. And then Jocelyn will be following with histology, HLA and serology standardizations, and other general clinical trial pitfalls. And again, this is really the title was really framed around regulatory expectations and by default sort of drug development. But I think these concepts are really applicable to any type of clinical research in celiac disease, and hope they will spurn some good questions and conversations. So, starting with patient-reported endpoints, always a little bit of a thorny discussion. So, just wanted to start with how the FDA defines clinical benefit, and so it's pretty straightforward. It's a positive, clinically meaningful effect of an intervention. A positive effect on how a patient feels, functions, or survives. You hear that feels, functions, and survives quite a bit. It's kind of a catchphrase. But it's pretty straightforward. It's, can be either how long a patient lives, so something that makes you live longer is obviously a good thing, or something that makes you feel or function better in daily life is either a good thing. And this can be demonstrated as either a comparative advantage in treatment of a disease or condition. So, one treatment is better than the standard of care, or comparative reduction in treatment-related toxicity. So, a new treatment is better tolerated than the standard of care treatment. Um, and further, I think it's important that the clinical benefit is described in labeling in terms of the population and outcome of interest measured. And I have one example here, which I think is a good one, the Xolair omalizumab food allergy indication, which I think bears a lot of similarity to labels that we might imagine in celiac disease in the future. But you see it's indicated for IgE-mediated food allergy in adult and pediatric patients, so the disease and population. And for the reduction of allergic reactions, type 1, including anaphylaxis. So, who it's for and what it does is the key part of the label. So, whatever you do in clinical trials are should be based around what you want that label to say, because in the end, it's what clinicians will be able to prescribe for, and and how the main benefits are often described to patients. And again, this is another interesting thing with this one, just like I think most celiac therapies, this is to be used in conjunction with food allergen avoidance. Celiac therapies would likely be mostly the same to be used in conjunction with a gluten-free diet. So, um, so survives is easy, obviously, and we don't talk about that much in celiac disease. Fortunately, we are not looking at, in general, survival benefits. Um, so we're really talking about ways to measure feels and functions. Um, and this clinical outcomes assessments is the broad range of how people feel and function. These can be the most common we talk about patient-reported outcomes, things like surveys of symptoms. Could also be clinician-reported outcomes, observer-reported outcomes, or performance outcomes. The sort of a classic performance outcome is like the uh, a 3-minute walk test or stair climbing or something like that. Um, so you can also do this theoretically through biomarkers, but that's a challenge. And it's what's interesting, and we often forget. So this came directly from an FDA document. A biomarker is not an assessment of how an individual feels, functions, or survives. What a biomarker is, it's a defined characteristic measured as an indicator of normal biologic processes, pathogenic processes, or response to exposure or intervention. And they have these examples: molecular, histologic, radiographic, physiologic characteristics. Um, and while theoretically, as I say, the FDA does accept biomarkers as a way to potentially measure fields and functions, and you see histology there, and it's easy to get excited and say, well, why can't we then use histology as the main basis for approval in celiac disease? Um, and as much as that continues to be an issue, because obviously intestinal damage is a hallmark of celiac disease and an incredibly important thing to manage properly. So um, I wanted to explain why there is a very high bar for biomarkers as a basis of approval. And I'm going to do that with an example that comes from pretty far away from celiac disease and GI in general. Um, so we're going to heart attacks. So here's two facts you may remember from residency or whatever. Ventricular arrhythmias occur pretty frequently, about 10% of patients post myocardial infarction, usually in the first week, mostly in the first 30 days. And also, ventricular arrhythmias account for 30 to 50% of 30-day mortality post MI, with things like thrombosis accounting for most of the rest. So, seemed like a very rational assumption that preventing arrhythmias will lead to a reduction in mortality, just the same way we presume that improving intestinal damage in CLTs will lead to improvements in long-term patient outcomes. And this was basically dogma for many years. And you even had really, um, you had a lot of articles like this that were now people regret having written, things like second generation antiarrhythmic agents. Have we reached antiarrhythmic nirvana? Because they were well tolerated and really good at suppressing ventricular arrhythmias post MI. And things were labeled in a lot of people. These were basically standard of care post MI to get one of these. But they hadn't studies to look at mortality had to be very large, very long, so people didn't really do them, and they weren't part of labeling. Enter the early 90s, the CAST study, cardiac arrhythmia suppression trial, where finally they did this huge study, 1,500 people for a year and a half, post-MI, looking at placebo versus the second generation antiarrhythmics. And indeed, you see a really significant difference in mortality between the two arms. This was evident really quickly and only expanded over time. What was sort of unfortunate is this part, is that the placebo were the people who did much better, and the people on antiarrhythmics did much worse. And again, it's hard to get an estimate, but the assumption is tens of thousands of additional mortalities were caused by this really logical assumption that a biomarker, arrhythmias on EKG post MI, could be used as assessment of benefit. And so this, along with a few other things, really put a really like threw a bunch of cold water on the idea that biomarkers, without a ton of evidence, can be used as evidence for benefit of a drug. And I think we're still living with the repercussions of this in the way the FDA really thinks about biomarkers. So it is a high bar for a reason. And until you really show that you have to do one of these studies showing that, intestinal improvement leads to something really good, until you do that, intestinal improvement with a drug, you will be stuck with feels or functions in celiac disease. We're also not alone in celiac disease. So this is just a rough graph. PROs used to be a pretty uncommon thing, and now they're basically standard in every FDA submission. They're not always the primary basis of approval, but they're often supportive. Whether you're talking about oncology trials or cardiology trials or whatever, almost no studies get done without some sort of PRO as at least supporting evidence these days. Um, so, as fields and functions came to replace biomarkers, the FDA expectations evolved. We had 2003, some initial framework, just giving some people some rough ideas of how PROs should be developed and validated. This was made much more comprehensively in 2009. 2015, they first published their formal PRO endpoint qualification pathway. And then 2019, they added a lot of information on how to do this well using new ePros and digital collection and real world data and mobile phones. Before that, it was all paper. And then most recently, just last year, they had some additional guidance, which was pretty interesting for some of us who follow this, use it about adaptive trial designs and really real world evidence integration into PRO assessments, how you can potentially stick these into doing pragmatic trials, putting these into your EMR, and using that to support an approval of a drug potentially, or for any other reason, label expansion, post-market assessments, lots of things. So the FDA has done a ton of work on this. Some of it has been a lot of it's been very helpful. Some of it has been challenging to implement. But again, I think versus actual diseases, in terms of what the FDA actually wants for PROs is quite clear. It's a long and expensive process, but it is clear. So what are these? So the main things that the FDA is looking for in a patient-reported outcome, they want to start with a conceptual framework. So did you talk to a bunch of patients about what's really meaningful in a PRO to help design it at first? Then how you develop the instrument. Did you refine it? Did you make an initial PRO based on that patient input and then go back and forth with patients and refine questions, eliminate questions, add questions? Did you do appropriate psychometric validation? That's reliability and responsiveness. Did you establish a clinically meaningful, important difference? You need to be able to package all of those first four steps into your regulatory submission package. So this is a ton of work. It's nothing that any single human can do. And even large companies usually have a lot of vendor support from companies specialized in doing this to be able to put all these pieces together in a cohesive package, because it can be very technical and often run into the hundreds, if not the thousands of pages for these submissions. Which is why it's important to start early, if that wasn't already obvious. Fortunately, we're in a pretty good place in the celiac disease. We have a couple of PROs, the celiac disease symptom diary, the celiac disease PRO, that have both been used in phase 2 trials. Both have acceptable psychometric properties. So really, we're sort of in the sort of phase 2, phase 3. We're at the top of that clock. But yet we won't really know until we hit that, blue and green, the regulatory submission, and really even post-marketing to see if, the things we've measured in clinical trial actually translate into real-world clinical benefit. Skip one, no. So, again, I said a little bit of this, but just a little bit more detail into, if you're looking at a PRO that you want to use in a trial, or if you're looking to develop one, Does it measure the outcome of interest, right? I mean, you can, when you talk to patients and do these intervals, lots of things will come up as parts of the burden of disease. I mean, for celiac disease, a key one is, anxiety around food exposure. That's a key, it's a very important endpoint. It may not be what you want the PRO to measure if you're looking at symptom improvement. Is it well defined and reliable? Is it appropriate for the target population? Um, is it appropriate for the target indication, right? It's not just who it's for, but what it's for. And again, content validity, reliability, responsiveness, all of these, and they're all spelled out in a CFR, are what the FDA is going to look for, and really need. And it's as much about documentation of this work as it is about actually making sure you have a good PRO at the end. So one question that is often asked is how much improvement is meaningful? And of course, that's indication, disease, and population specific. But there are some kind of rules of thumb that you can see across a bunch of trials. So symptom improvement, generally 30% improvement, reduction in symptoms is generally clinically meaningful and would support something useful. That's consistent, whether you look at IBD trials or IBS trials, it's sort of, for instance, are kind of the same. Functional status, walking or whatever, 25% improvement. Quality of life, a 20-ish% gain. Patient satisfaction, one we don't really talk about a lot, but can be important, especially if you're looking at, moving from IV to sub-Q or something like that. But there you want a larger gain because you're really not affecting the symptoms, functional status, things like that. So you really expect sort of a larger improvement if patient satisfaction is really your key, the key thing you're looking for. Um, so just to also state that this is an art as much of a science as a science. There's no perfect clinical outcome assessment. And clinical outcome assessment strategy. You're always either going to miss things you wish you captured, or you're going to have a questionnaire that's so long that people don't do it, and compliance goes down. So I think that's always the first thing to think about very critically is, how do you balance comprehensiveness with patient burden and feasibility? Um, Also, how does what you want to do compare to what else has been done? And number two, differences in PRO, there might be something a little bit better for your trial versus another trial, but then does that make it harder to compare your results versus whatever else is published in the literature? Um, Diversity is obviously critical and really important in any of the studies we do, but that adds time and development costs in order to adapt the instruments. And some of them will be very hard. If it's something around food preferences, that's going to be very challenging to do across a diverse population with different food preferences and cultural norms. Um, and something that we worry about a lot, and there's different ways to handle it that are very statistical, and I won't go into them a lot, but is incomplete responses. You're never going to have perfect data in PROs. There's always going to be missing items, missing days. Somebody's going to forget. ePro is going to stop working. They're going to travel, and the time zone is going to mess it up. So thinking about how you're going to handle that ahead of time, how much data is enough to make a conclusion, how much when you need to throw out that data for the week or even for the participant, though hopefully not. What kind of sensitivity analysis you're going to do. All of that is really helpful to think out ahead of time because it's inevitable. I mean, again, there's no such thing as perfect when it comes to PRO completion over a trial. Um, So, just building off that a little bit more, you do what you can to keep the PRO completion as robust as possible. That starts with patient recruitment. Are the patients, not only do they have the disease, which I'll talk about in a second, but are they actually able to complete the PROs? Are they committed? How can you keep them committed? Can you gamify it? Whatever. So, um, choosing the right patients who can actually do the trial, the limiting the timing of the collection to what's clinically relevant. Don't, more is not always better. Um, does the technology actually work and is it convenient? Um, and then it's, do you have a way to monitor it? I mean, it's never fun when you're at the end of the study and you realize that only half the people have completed the PROs. You really want to be able to monitor that real time. And fortunately, as we've moved to electronic collection systems, that's much more feasible, much more possible. Although I think the systems to do that are still not generally very user friendly, and they can be a little burdensome. But this is really helpful. Not only to course correct if something's going wrong, but it's part of the regulatory package of showing compliance with best practices. So with that, I'm going to move off of the PRO section. I know that was a lot of information, but I want to go to another topic, which is of great importance, is clinical patient trial selection. And again, this is I'm taking this largely from a lens of drug clinical trials, but really is I think broadly applicable. Um, and so obviously this matters for everyone, right? It doesn't help anyone to put the wrong patients in your trial because it's bad for the patients, it's bad for the trial. You're not going to achieve your research goals. So there is some things we've talked about a lot, which is these assessing celiac disease activity appropriate for trial goals. So in studies assessing improvement in active celiac disease, are ongoing symptoms really related to gluten and celiac disease? That's a really tough one. We've talked about that a lot in this series and other series. And as far as I know, there's no great answer to that. The other one, which is coming up more, is in studies assessing protection from gluten exposure, a gluten challenge study where you're looking typically at exacerbation of symptoms, exacerbation of histology. How well controlled should patients be? How normal does their VHCD need to be? And there may be sort of benefit, there may be a ceiling and a floor effect for that in both directions. So that's certainly something to consider. But I really want to talk a little bit more about exclusion of patients without celiac disease. This is something that I think has been an issue in all celiac trials and maybe is not something we've talked quite as much about as we should be talking about. So, so again, does this patient really have celiac disease in a clinical trial? Is this an under-discussed issue? I think it is. So it's hard to find good data on this, but this is sort of my personal experience, is that about 10% of patients are excluded from trials due to HLA DQ2, DQ8 negative. So that's why that should always be part of your screening for patients for celiac disease clinical trials. And so the assumption obviously is those patients do not have celiac disease, but it doesn't really help how many of the others, if 10% of patients being screened, we know for a celiac disease trial, we know don't have celiac disease because of HLA. How many of the other 90% also don't have celiac disease? Um, and I want to give an example of a patient. This is something that came up recently for me. So here's a patient. You can see the data. You have a borderline positive tissue transcutaminase IgA. You have a negative EMA, and you have, a normal D2 and, some patchy changes in the duodenal bulb. So per most trials that want biopsy confirmed serologically positive CLTs, this patient would meet inclusion criteria. Um, I don't think there's an easy way to pull this. I wish there was, but hopefully patient in a trial would make you nervous because it would definitely make me nervous. And if this is a patient I was seeing in clinic, I would say, well, let's see how you feel. You're welcome to try gluten-free diet, but I'm honestly not convinced. And I would probably, if they were feeling fine, I would say, let's just keep an eye on things, honestly, and recheck your celiac serologies in another year or 6 months or whatever, or as needed. But these are patients who are very easy to include in clinical trials, although they're probably not the ideal patients we want in clinical trials. So with that, I want to actually first give thanks. These are slides that were provided to me by Jordan Doctorow. Many of you know him. He works with Beyond Celiac and he's a very established clinical developer in neurologic disorders. And he after having, if you think it's hard to figure out who has active CLTs in our trial, you can imagine neurologic disorders is very hard to figure out who has active disease and what status there are and where so many diseases overlap. So he formed a company and he shared these slides with me to share today, which I think are a very helpful way to think about this, um, about how to really make sure we're getting reagent grade patients in our clinical trials in a much more uniform way, especially in larger multi-center trials. Um, so and really this revolves around this concept of an enrollment authorization committee. So these are a group of experts who are not otherwise part of the trial. They're not PIs, who basically say yay or nay to any patient who's being screened for the trial based on their objective information. This really can help to standardize enrollment and exclude patients who are sort of borderline. Um, and so the theory behind this is that, there is an important and rational difference between medical care versus clinical research. So in medical care, we do individualized therapy, what's best for the patient. We use flexible judgment. We care for everyone who walks in the door, as best as we can, often in time with some diagnostic uncertainty. And really, it's a physician-driven, or I would say physician-patient-driven activity, practice patterns, anecdotal experience, personal preferences for both the patient and the physician and the clinician. Um, Clinical research, however, is different. It's protocol-driven. Um, there's unknown risks. That's why we're doing the trial. There's clinical equipoise. We have to do informed consent. It's a much more rigorous approach with strict exclusions. And in here, the research protocol drives activity. So it's not about necessarily what the physician thinks of this person probably has celiac. It's really, does this patient really have celiac that meets the standards defined in the protocol? And including what, when, where, why, and how they've been treated in the past and their disease status. Um, So research candidates should be medically stable. Um, they should have social support in place. Again, going back to that feasibility, willing and able to follow the research protocol, which is sounds easy, but is not always immediately apparent. Uh, both to the patient and to the clinician about, especially as it's not always clear what they're getting into, how rigorous participation can be. And the goal is, again, reducing the variability and error and increasing the signal to noise ratio by really getting those patients who are best able to go into the protocol and are most appropriate for the protocol. Um, so this is really, so these enrollment authorization review committee rationales is really around suitability. So it maximizes the entry of candidates most likely to provide high quality interpretable data, minimizing variability, reduces risk of dropout, ensuring correct diagnosis, um, keeps an eye out for unusual or inconsistent screening data, um, data considered in aggregate. So this groups of clinicians get tons of experience, and with priority placed on really finding those suitable things for predominantly for medical on the medical aspect, but also potentially behavioral and social, depending on the type of trial. Um, and also it allows for upscaling, uptraining of the sites, especially if you're working with a lot of clinical trial sites who haven't done a lot in celiac disease, the ability to engage with these peers or see the decisions these sort of experts in clinical care and research can make about the stage and really help them screen better for the current trial, but also future trials. Um, So this has been used again a lot in neurologic disorders and is FDA and other agencies are very okay with it, are actually very supportive of this approach. But this was an example that he gave me of an independent committee of neurologists with Parkinson's disease expertise with a charter. And again, all the things I said, they ensured correct diagnosis, data made sense, entry criteria, um, and and again, when questions arise, it is not like a no, you screened the wrong person. It's really a discussion and a collegial interaction, and hopefully a time to and uh, an opportunity to educate more broadly on the disease in progress and under study. So with that, I will end, but but I think these are things that we should be thinking about as well, that everyone should be thinking about as they design and execute their clinical trials. So with that, thank you, and I'm right on time to pass it on to Jocelyn.
Thank you, Dan. I am going to talk a little bit about the other side of the co-primary endpoint, which is histology, and also a little bit about what Dan has already alluded to, the fact that unfortunately clinical trials are studies of humans by humans. So to start off with, Dan, um, what is the FDA looking for in a celiac disease clinical trial? And they're really although, and this is from the FDA draft guidance, but I think it's important to remember that that was 2021, and hopefully the FDA's thinking has evolved somewhat since then, and that that's a particular indication for celiac disease treatment. But with those caveats in place, really what they want to see is feels, functions, and survives, which is in practice signs and symptoms, which is why we've talked about PROs. And then they're concerned about celiac disease as a mucosal disease and the idea that there has to be some assessment of end organ damage, which is where biopsies really come in. And the FDA really favors a co-primary endpoint. So they would like to have both of these in the same trial and not one trial powered for symptoms and a separate trial powered for inflammation. Next. And the rationale for that is that maybe neither of those endpoints are very good, but if we mix them together, we can get something better than either one alone. And so symptoms are subjective, and as Dan alluded to, it's not always clear if symptoms are celiac disease or something else, but they're important and we're going to need to include them. Also, we know that often the people who want to volunteer for a trial are the ones who are less symptomatic, particularly if the trial involves large amounts of gluten. And so if you're starting with asymptomatic people, having everybody hit a symptom endpoint might be somewhat difficult. Um, the other reality which we face in clinical practice and in clinical trials is that there's not a clear correlation between what the biopsy looks like and how the patient is feeling. And that's why biopsies alone are not sufficient to approve a medication. And so the idea is if you have this co-primary endpoint, it's going to benefit the patient and you're going to have some idea of what's happening to disease activity. And so getting into the nitty-gritty of what does a co-primary endpoint mean. And I think this is a really important point that we sometimes gloss over, which is, first of all, both endpoints must be meaningful and they must also be sufficiently powered within the trial. And so having a co-primary endpoint may drive you to have a larger trial because you have to have enough patients to meet your symptom endpoint as well as your histology endpoint. And in order for the trial to be considered a success, both endpoints need to be met and they have to pass the significance test. But what's important is you have to have enough patients who have an improvement in symptoms and enough patients who have an improvement in histology, but they don't necessarily have to be the same people. And this is where there's clearly scope to have people who are less symptomatic in a trial with a co-primary endpoint that includes symptoms. And of course, this makes life more statistically complicated, which is why it's important to have professionals, and I'll say no more about that. Um, and so how did we get to this point of co-primary endpoints? I think part of it is biologic endpoints are not always the most robust assessment. And we don't want to get too far down the road before we realize actually how this drug affects these two endpoints is discordant. And so having them both in a single trial helps to make sure that both are moving in the right direction. The disadvantage of this is, of course, sample size, complexity, and then what do you do if you meet one of the co-primary endpoints, but not the other? And I think that's something that we will have to sort out, um, hopefully later rather than sooner. Next. And so when we think about celiac disease, we're collecting biopsies. And I think we think, oh, well, we need a biopsy, and we don't necessarily think much past that. And this gets into the idea that we're doing things, but this isn't clinical practice. And in a clinical trial, we really want to be rigorous and we want to be consistent. And in a sense, a clinical trial, particularly a multi-site clinical trial, is a massive exercise in delegation. And so it's really important to make sure that it's clear what is expected and what is required. And so a biopsy, there should be some definition of where to sample from, how many samples to get. And then we all know in the laboratory that you're going to have pre-analytical variation that's going to affect your outcome. And we can control that in clinical trials just like we can in the lab. So it's important to think about how to manage all of the processing of the biopsy before you even get to looking at the biopsy or thinking about what you're going to do when you look at the biopsy. And I think this is an area where we're working on figuring it out for celiac disease, and making sure that we get it right means that we'll get the most value out of the trial. Next. And so now we have a biopsy, it's hopefully oriented, and we have somebody looking at it. And the sort of standard now is to have some sort of quantitative endpoint. I think there's some disagreement about what that might be, and to have the review blinded, ideally centralized. And we talk a lot about variability in pathologists clinically, but in clinical trials, we really don't want to have variability in our pathologists. So if there's multiple readers, it's important to have some training and to continue to check to make sure that your readers are interpreting the same biopsy the same way, not only themselves in terms of inter-observer variability, but also if you give it to any of the pathologists on the trial, you're going to get a similar result, because this is a really important outcome where we're really looking at decimal points. And these sources of error can really make it harder to understand what's going on in the trial. And of course, it's always good to go back in time. So ideally take images of the biopsies and have documentation of how vellus height and crypt depth, for instance, were measured or which IELs were counted. And we all know that one of the problems with biopsies is that it's a 3D structure that we're looking at in a 2D frame. And so we're not always going to get the cut quite right. It's not always going to be on the right angle. And it's important to have a systematic way to make sure that the pathologists are at least starting with material that's interpretable. Next. And so I didn't leave much about biomarkers because, at least from an FDA perspective, they're nice to have, probably important, but they're not going to get you that FDA labeling that you want. And so they're really seen as something that's appropriate for a secondary or exploratory endpoint and can really be supportive to provide additional evidence that there is a biological response or that that biological response is real. I think in celiac disease, we actually have another application for the biomarkers, which if we're taking people who we expect are on a gluten-free diet, or if we're taking people who we're expecting to have gluten, we can actually monitor how much gluten they're getting as a biomarker, which can also be very helpful, as we've seen in interpreting trial results, because people don't always do what you ask them to do. And I think a good example of a biomarker would be something like IL-2, which can be very helpful for tracking immune activation, but is not actually histology and isn't going to serve as a proxy for mucosal damage without more validation. Next. So just like biopsies, just like PROs, biomarkers require rigor in order to get a good result. And there's lots of ways in the implementation that we can make mistakes that are going to make the data harder to interpret. And so particularly with serology, we know there's different assays, we know there's different thresholds for cutoffs, and we really have to be cognizant of making sure that we're all talking the same language or using the same thing. And reproducibility is really important. And there's lots that happens between the patient and the study team and the CRO and the actual analysis of whatever you're looking at. And those are all steps where things can go wrong to differing degrees. I was joking with Dan that we should really just give people examples from personal experience of all the things we've done that have gone wrong, but he assured me that nobody would really want to sit that long. So I think it's important also when you're thinking about biomarkers to really be thoughtful about what the biomarker is for and make sure that the biomarker matches the kinetics of what you're trying to measure. So something like IL-2, we're thinking about acute gluten exposure, GIPs, this is a longer window, serology is a longer window, and histology is probably a much wider window, and really how much gluten you're giving people is equally important there. Next. So this is where I wanted to end it, which is just this idea, which I think the more I get involved in trials, the more I realize that this is really the crux of the issue, and that I would be the worst possible clinical trial participant ever, is that trials involve real people. They have complex biology. They have other things to do with their life other than the clinical trial. And I think this is really where often the perfect clinical trial comes up against finding a participant who can actually participate in that trial. Because if the windows are too narrow, if there's too many assessments or the assessments are too burdensome, then it's hard to get all of them. And sometimes you don't end up with something as good as if you've been less ambitious in designing the trial in the first place. Um, I think it's also important to remember that both the investigators and the study site staff and the participants are human, and they all have behaviors that are modifiable. Um, and that everybody is making judgments all the time, which may or may not be covered in the protocol. And so I think this is where, if you want the next slide, Dan, it's important, particularly in celiac disease, where there's lots of behavior that's inherent in the trial that's crucial to everything that we're measuring. And it's not really standardized. We just ask people to follow their usual gluten-free diet. What does that mean? And how are we going to monitor it? And um, and if we're getting PROs, what are we going to do in order to make sure that people actually fill them out? And um, if there is something that happens in terms of an inadvertent gluten exposure, are people allowed to take medications? What are they allowed to take? Recognizing that things are going to happen outside of the trial that affect the trial. And I think it's helpful to have a plan in advance so that there's some consistency on how those things are managed, and particularly if there's a lot of gluten and there's a placebo group, thinking about how to manage gluten symptoms in the placebo group in a consistent way to make sure that the data is interpretable at the end of it. So it might be better to have a flexible gluten dosing protocol that everybody can get through than have lots of people dropping out. Also, I don't think we think that much about what we're actually given or what we're actually doing, starting with when we're actually doing a study visit, doing a trial, what the actual kits look like. But there's lots of environmental controls that can be put in place here that are going to ensure that data collection is more timely, it's more accurate, it's actually happening. And I think it's as important as thinking about how you're going to orient your biopsy and what you're going to do with it to make sure that it's really clear and it's really hard to go wrong in terms of gathering the measurements that are needed. And that was all I had, anticipating there may be some questions.
Yes, Dr. Leffler and Sylvester, thank you so much for those excellent presentations. Excuse me, just want to open up the floor to any questions there might be right off the bat.
I can start with a question. That was great. Thank you to both of you for your presentations. Regarding the PROs, what is sort of an expected compliance rate in clinical trials, and are we seeing that in the celiac disease clinical trials that the PROs we've chosen are completed and seem reasonable and aren't too burdensome? And then also, when we think about some of the trials, we're almost doing an inverse, right? We're like giving people gluten, and so we would actually expect that those people with placebo and gluten, their PROs might get worse, and then others might get better or stay the same. So we're looking for like a 30%, still a 30% difference and a 30% worsening almost in the placebo group when we think about that.
Yeah. So those are both good questions. So in general, if you can get most, you can consider like, oftentimes it's common to do for a daily PRO to look across a week and average that out. In general, if you get 5 days out of the week, you generally consider it an evaluable week. Some studies go down to 4 days a week. So just given that, if you can get on average 75%, ideally above 80%, you're doing pretty well. And that's generally all you can expect. And that's totally sufficient. So, in general, if we can get people down to missing, 1 day a week, um, of their daily PRO, that's pretty reasonable. It is harder though if it's always the same day. Like they're always going out to eat on Sunday, so they're not doing their PRO. Those are the days we most want to capture. So if it's sort of more of an unbiased, forgetting once a week, that's more acceptable. But in general, if we can get above 80%, everyone's generally pretty happy. Ideally higher, but that's a sufficient level. And yes, that's a really good question. The vast majority of studies in the literature are on improvement of symptoms, right? We're sort of in a unique, it's us and food allergy and a few others that have the potential to do these challenges where we're doing worsening. I think those, so I don't think there's a benchmark in the same way. And I think that work is still yet to be done on how much difference of, protection is clinically meaningful. My guess is it won't be that different, but again, I think we need to run those studies to really know. But yeah, it's a good question.
And I think PROs are a good example of a human behavior that's really important for clinical trials. And so it's really important that the participants know why those PROs are so important and that they really need to fill them out if they actually want a drug for celiac disease, and that, it's an important measurement.
So this is maybe a little bit off topic of exactly what we're supposed to cover today, but I'm wondering how all of this around endpoints really eventually translates into labeling and coverage, particularly for the patients who are considered like very well controlled and are looking at the drugs for more like quality of life and like reducing anxiety burdens.
And you want to do that? You want to take that one?
I was
Or?
going to let you take it, Dan.
I mean, so I think there's a recognition that those are really important facets of life with celiac disease, with a lot of disorders, but celiac disease in general. And if we look to the food allergy, while anxiety and caregiver burden and things like that were not, where they were not what we call the primary basis of approval. That wasn't what checked the box. They were actually really important for coverage, especially in Europe, interestingly. That was one of the main things that NICE, the UK group, looked at and said, all right, this is something that we should be paying for because it's not just about the prevention of that, 1 or 2 food allergy episodes a year. It's about all the other days and all the other stuff that happens around that makes it a cost-effective therapy. So, to me, I think I look at it, I think I'm trying to, let me know if I'm not answering your question at all, but I think, these things are really about, understanding the societal effect of a therapy, because again, we're asking the society to pay for the therapy in general. It's not just the patient. So, I think these are really important things that we need to think about how best to capture. Do we need specific PROs? Can we borrow them? How do we study that? Is that more of an open label than a randomized controlled trial? Even if it's not what the FDA is saying, yes, that's what you'll need for approval. I don't think it's. I think we really ignore those things at our own peril.
Which means more PROs and more surveys.
More, yes, this is a balance, right? Yeah, and again, you put so much time and effort into a study. You hate to you're like, oh, I want to get one more thing. I want to get one more thing, and then suddenly you have like 30 more things, and it's not good for anyone. So it is a balance.
Vanessa, does the CDF have, I know in this call I was on yesterday, there was talk about what can we develop for patients to help them like be more compliant and understanding of clinical trials? Um, and not just for CDF, I guess, but does any service like kind of sit down and or give some kind of information about why the PROs are so important, and even though they're cumbersome, this is why you need to do it and what we're looking for? Or do patients just get these surveys and then we just nag them to do it when they're not doing it?
So, at least for clinical trials that our foundation recruits for, we have a whole call center that every patient goes through, and they have these conversations with them as they're talking to them about enrolling in a trial. And so it's something that we definitely address. I don't know that we've ever done like a specific like class for like a broader group of people, but it's something that is addressed on every phone call with everyone that signs up through us.
Great point. It does take a village, right? And I think the more we're all sort of saying the same things and working to sort of educate, the better off all these trials will go. And I think that that also goes to the clinician side about who's appropriate and who's not. So,
All right. Well, while there's a pause in the questions, I'm just going to share here really briefly. For those who might be tuning in discontinuously, or even if you're tuning in now, um, we actually have a means of claiming CME credits. So you can just follow these instructions here. You can text this number and you'll get instructions on how to claim those. But I don't want to belabor that too much in case there are any other questions for our speakers. All right. Well, it looks like we're coming up on time here anyway. So that being said, thank you, Dr. Leffler and Silvester, so much for your time and for this excellent talk. And just also excited to share for those tuning in. We do have some upcoming webinars. We have a college summit coming up on the 16th of April. And then after that, on the 12th of May, we have building a celiac center with capacity for trials. So building on this very important topic of celiac disease research and how to conduct some of these prospective studies. And all these webinars are recorded and posted on the SSCD website for members. So with that being said, feel free to log in and watch at your own leisure. And thank you again to the webinar committee and SSCD as a whole, and of course our speakers. And thank you for tuning in, everybody.
thank you so much.
Thanks.
Everyone.